Assalamu alaikum, my dear students. I hope you all are doing good. In this lecture today, we are going to continue talking about the membrane transport. And this lecture is particularly going to be about the active transport across the cell membrane. We have already discussed in the last lecture about the passive transport across the cell membrane. If you have not watched that video yet, please go and watch that first. So this lecture is going to be about the active transport. We've talked in the passive transport that in passive transport there was transport across the cell membrane, but there was no utilization of energy for the transport, be it the simple diffusion or be it the facilitated diffusion. We have seen in facilitated diffusion there are membrane proteins utilized for the transport, but those membrane proteins were not utilizing ATP for the transport of materials. But in this active transport, which we are going to talk about today, in active transport, there is definitely going to be the utilization of ATPs. And this active transport is actually going to be uphill transport. Passive transport was downhill transport. That means that was the transport from high concentration to low concentration, but this active transport is going to be the transport from low concentration to high concentration. We also call it as the uphill transport. Now, this active transport is actually divided into two types. There are different mechanisms. There are different examples we have to talk about in this lecture. But broadly, our active transport is going to be categorized into two types. One is I have mentioned the name there. One is the primary active transport, and second is the secondary active transport. There will be utilization of energy in both the mechanisms, but what will be the differences? Let's find out. So let's talk about the primary active transport first. So primary active transport definitely अभी जो मैंने आपको कहा कि primary active transport में there is definitely going to be ATP utilized. So that is the first thing. ATP utilized, and this ATP in the primary active transport is going to be utilized directly by the transporter protein. The transporter protein which will be involved in the transport, which will be involved in the primary active transport, will be utilizing the ATPs directly. So that is an important concept here. So there will be direct utilization of ATPs by these transporter proteins. Right. The second thing is, in the passive transport, we have seen that was the transport which I mentioned just earlier, that in passive transport, there was transport from high concentration to low concentration, and active transport is definitely going to be the transport from high concentration to low concentration. Right. So primary active transport is where the ATPs will be utilized, and in particular the primary active transport is going to be where the transporter protein is actually utilizing that ATP, and that ATP is being utilized directly by that transporter protein. Okay, now let us talk about a few examples which actually follow the mechanism of primary active transport. The first one which we are going to talk about is, that is actually very important example. Whenever you talk about the primary active transport, this example should come first in your mind. The first example for this primary active transport is going to be sodium potassium ATPase. Sodium potassium ATPase. Now, what did I tell you in the last class is that sodium is in higher concentration outside the cell, and it is in lower concentration inside the cell. So if this is outside of the cell, this is inside of the cell, we say that the sodium is in higher, okay, let me write it here. Sodium is in higher concentration outside the cell, and sodium is in lower concentration inside the cell. And when we talk about potassium, potassium is in higher concentration inside the cell, and potassium is in lower concentration outside the cell. This you have to remember every time, that sodium is in higher concentration outside the cell and then potassium is in higher concentration inside the cell. Now, as the name indicates, it is sodium potassium ATPase. ATPase means it is actually an enzyme which is utilizing that ATP for this transport of sodium and potassium. And this sodium potassium is actually the example of primary active transport. So that means this transporter protein here will be utilizing the energy, will be utilizing ATP. It will be actually converting that ATP into ADP and inorganic phosphate. And it will be that energy which will be liberated from there will be utilized by this transporter protein for the transport of sodium and potassium. But this is the active transport. So definitely sodium and potassium will be transported against their concentration gradients. What does that mean is that sodium will be transported from inside to outside, that is from low concentration to high concentration, and this potassium will be transported from outside to inside, again from low concentration to high concentration. Now what this sodium potassium ATPase actually is doing, now suppose this sodium potassium ATPase, and you have to remember one thing, that sodium potassium ATPase is present in almost all the cells of the body. It has a great importance in our body. Different mechanisms are actually depending upon this sodium potassium ATPase. We are going to talk about that in many lectures which we are going to upload in near future, inshallah. So this sodium potassium ATPase, now consider it has these 5 arms. You consider it as 5 arms of the sodium potassium. It is actually 5 binding sites of sodium potassium ATPase. It is also called as sodium potassium pump. Now 2 binding sites are outside of the cell membrane and 3 binding sites are towards the inside of the cell membrane. Right. Now these 3 binding sites are definitely going to be for 3 sodium ions. So sodium will come and bind on these 3 binding sites, and 2 potassium ions are going to be binding on these 2 binding sites, which are actually towards the outside of the cell membrane. Right. So here is sodium potassium ATPase. It has 3 arms towards inside and it has 2 arms towards outside. The 2 arms which are towards outside are actually holding 2 potassium ions, and 3 arms which are towards inside of the cell membrane are holding 3 sodium ions. Right. Now what happens is, once these binding sites are actually bound to these two potassium and 3 sodium ions, what will happen is that this will actually utilize that ATP, and after utilizing that ATP, now consider it will flip around and it will send these 3 sodium ions out and it will send these 2 potassium ions into the cell. Now, what is the job of sodium potassium ATPase? It is very important because this sodium potassium ATPase actually, when we will talk about this secondary active transport, mechanisms are actually dependent upon the sodium potassium ATPase. That is very important. So the sodium potassium ATPase has a great importance. What it is doing is, it is sending 3 sodium ions out of the cell against their concentration gradient, and it is bringing 2 potassium ions into the cell against the concentration gradient of the potassium, because potassium is in low concentration outside the cell and sodium is in low concentration inside the cell. That is all about sodium potassium ATPase. This is the first example of primary active transport. Now let us talk about the second example. The second example is actually, it is called as the calcium ATPase. Calcium ATPase. Now this calcium ATPase, when we talk about calcium ATPase, it is not actually there in the cell membrane. It is actually in the membrane of a particular cell organelle. Because we know that cell membrane is a phospholipid bilayer, and the cell organelles, membrane-bound cell organelles, the membrane is there is also the phospholipid bilayer with different proteins there. Right. So one of the cell organelle present most importantly there in these muscle cells is called as sarcoplasmic reticulum. Right. So sarcoplasmic reticulum is actually, we can say it is smooth endoplasmic reticulum there in the muscle cells. Now, the sarcoplasmic reticulum, it has different functions in the muscle cell, and one of the important function of this sarcoplasmic reticulum is storage and release of calcium, because we know that calcium is very important for the contraction of muscles, for muscle contraction. Now that calcium which is utilized for the muscle contraction is actually stored inside the sarcoplasmic reticulum. And once it is needed for the muscle contraction, the sarcoplasmic reticulum will release that calcium into the cytoplasm. Now, let us suppose the muscle is done with the contraction part. Now this contraction has to be followed by the relaxation. Now, during relaxation, what we have to do is, we want that calcium to be taken back into the sarcoplasmic reticulum. Sarcoplasmic reticulum has released that calcium. Now, suppose here is the calcium which has been released by the sarcoplasmic reticulum, and that calcium has done its job for the muscle contraction. Now, what we want is, fine. So now what we want is that this calcium, we want to take it back into the sarcoplasmic reticulum for the relaxation part. Now what happens is, calcium in the cytoplasm is in low concentration, because this sarcoplasmic reticulum is the storage site of calcium. So calcium inside the sarcoplasmic reticulum is in higher concentration. Right. Now, calcium has to be taken from cytoplasm into the sarcoplasmic reticulum. That means it has to be taken from low concentration to high concentration. Now, for this transport, we have on the membrane of the sarcoplasmic reticulum, we have these special proteins there. And these proteins are actually calcium ATPase. And this calcium ATPase, what it is doing is, it is utilizing the ATPs directly, it is utilizing the energy directly and taking the calcium from the cytoplasm into the sarcoplasmic reticulum. Or we can say it is taking the calcium from low concentration to high concentration. Right. So this is the second example, which is actually calcium ATPase, and this calcium ATPase is particularly in the membrane of the sarcoplasmic reticulum, where it is taking the calcium from cytoplasm of the muscle cell into the sarcoplasmic reticulum from low concentration to high concentration. Now, that is all about the calcium ATPase. Now, the third example which we are going to talk about is actually called as proton pump. It is the third example of the primary active transport. Now, this proton pump, when we talk about the proton pump, there should be one thing which should come in our mind, that is our stomach. Our stomach contains a lot of HCl, hydrochloric acid. Now, that HCl is actually formed because we have a special type of the cell present there in the stomach, walls of the stomach, that is called as parietal cell. And that parietal cell is actually actively transporting protons or hydrogen ions into the lumen of the stomach. And then hydrogen ions or protons plus chloride ions, they form our this HCl or hydrochloric acid. Now suppose here is our stomach, and in the stomach, what did I tell you is, there is a parietal cell. Now suppose here is the parietal cell, and on the luminal membrane of the parietal cell, we have a proton pump there. Okay, and proton pump, what it is doing is, it is actually transporting the hydrogen ions or protons into the lumen of the stomach, definitely from low concentration to high concentration. Right, it is transporting the hydrogen ions from low concentration to high concentration, and it is called as proton pump. It has actually one more job to do. It actually it also transports the potassium ions from the lumen into the cell, that is again from low concentration to high concentration. So it is also called as proton potassium pump or proton potassium ATPase. Right. So it is utilizing the energy directly, it is utilizing the ATPs directly, and it is transporting the protons from inside of the parietal cell to outside in the lumen of the stomach. Or we can say it is transporting the protons from inside to outside of the cell membrane, from low concentration to high concentration. Now, this proton pump is very important because there is a drug which works on this particular protein, that is proton pump, and that drug is going to be very straightforward name for this, that is going to be the proton pump inhibitor. Yeah, proton pump inhibitor, actually we have that drug pantoprazole, for example, that is actually working on this proton pump, and that actually inhibits the proton pump, hence inhibits the or decreases the secretion of these protons or hydrogen ions, so that we can decrease the formation of HCl there in the stomach. Right. So we have the third example of the primary active transport, that is proton pump, and that proton pump is actually most importantly there in the stomach, that is on the luminal membrane of the parietal cell there in the stomach. So that is all about the primary active transport. Let's now talk about the secondary active transport. So secondary active transport is again the transport which will be actually utilizing the ATPs. But the difference here is that the primary active transport was utilizing the ATPs directly. That means the transporter protein which was involved in the primary active transport was utilizing ATPs directly. But here in the secondary active transport, ATPs will be utilized, but they will not be utilized directly by these transporter proteins, which we are going to talk about. Right. So the first thing is about the secondary active transport, that there is ATP utilized. Just like the primary active transport, there is utilization of ATPs, but there will be indirect utilization of ATPs. Indirect utilization of ATPs. Right. So there is indirect utilization of ATPs. Now the second thing is that there is actually going to be more than one substance which is going to be transported in the secondary active transport. Okay, now suppose here is suppose there can be two substances which can be transported, there can be three substances which can be transported. Okay, there can be substances transported in the same direction, there can be substances transported in the opposite direction. We are going to talk about all these in these examples of the secondary active transport. Now suppose there is substance A which is going to be transported. Now this substance A is actually, you have to remember this, that this substance A is actually going to be transported from high concentration to low concentration. Don't get confused. We will talk about this, the secondary active transport, because there is definitely the transport from low concentration to high concentration. But remember that there is more than one substance is being transported. So first substance which will be transported, suppose substance A will be transported from high concentration to low concentration. High concentration to low concentration. And remember that the substance A is almost always going to be sodium. Okay, it is mostly it is going to be sodium, which will be transported from high concentration to low concentration in the secondary active transport. Right? So if sodium is being transported from high concentration to low concentration, very simple, sodium must be transported from outside to inside of the cell. Right. Now, there will be another substance, for example, substance B. And this substance B will be transported from low concentration to high concentration. Right, from low concentration to high concentration. Now, in order for this secondary active transport to work properly, we must understand that there should be that sodium potassium ATPase we have talked about there. Right? Now suppose here is here is a cell, here is a cell membrane, here is that sodium potassium ATPase. What that sodium potassium ATPase is doing, it is continuously sending 3 sodium ions out, and it is continuously bringing 2 potassium ions in, right? That we have talked about as the first example of primary active transport, right? Now, if this sodium potassium ATPase is continuously sending 3 sodium ions out, what does that mean? Inside of the cell, the sodium concentration is going to be decreased, right? So inside of the cell, the sodium concentration is decreased because there is a sodium potassium pump which is continuously sending 3 sodium ions out of the cell, making that concentration gradient, making the inside of the cell, making the sodium concentration inside of the cell very low as compared to outside of the cell. Right. So one more time, we have sodium potassium ATPase. Sodium potassium ATPase is not the example of secondary active transport. I'm just revising that what we have talked about in the primary active transport, because it is the sodium potassium ATPase on which our secondary active transport is actually depending on. Right. So sodium potassium ATPase actually makes sure that there is low sodium concentration inside the cell and there is high sodium concentration outside the cell. How it makes sure? Because it is continuously sending sodium ions out of the cell. Right. Now, what if this sodium potassium ATPase is not working? We will not be making the sodium concentration inside the cell decreased. There can be sodium concentration inside the cell, which can increase because we are not sending the sodium ions out of the cell. Now remember that. Now we have to talk about the different these examples of secondary active transport, and the first example we are going to talk about is actually called as sodium glucose cotransporter. What did I tell you? That there will be more than one substance which will be transported in secondary active transport, and one substance will be definitely from, at least one substance will be definitely from high concentration to low concentration, and that other substance will be definitely from low concentration to high concentration. Now, the first example is actually going to be the sodium glucose cotransporter. We call it as SGLT. Okay. Now, SGLT, and this is sodium glucose cotransporter. This sodium glucose cotransporter is best example for this is there in the nephron. Right. हमारे nephron में what happens is that nephron is actually a basic functional unit of the kidneys, and that nephron is involved in the formation of urine. Now, formation of urine is completed in 3 steps. One is the filtration, then there is tubular reabsorption, then there is tubular secretion. Now, when we are filtering the plasma, now suppose here we have a Bowman's capsule there, and that Bowman's capsule is actually where there is filtration taking place, and after Bowman's capsule there is kidney tubules or renal tubules, and the first one is proximal convoluted tubule. Right? The first part of the renal tubules or the tubules of the nephron is proximal convoluted tubule, which is right after the Bowman's capsule. Now, what happens is, here in the Bowman's capsule, we are filtering plasma, and there are different things from the plasma which are being filtered except the plasma proteins. And the most importantly we have to talk about here is sodium and glucose. So definitely there will be sodium which will be filtered here, and there will be glucose which will be filtered here. Now, after being filtered, after this sodium and glucose is being filtered, what happens is we want this sodium and glucose to be reabsorbed back into the blood, because हम नहीं चाहते कि ये glucose और sodium जो है हमारे यूरिन के साथ लॉस हो जाए. हम चाहते हैं कि ये glucose और sodium जो है वापस हमारे blood में आ जाए, because we need that glucose there in the blood in our body. We need that sodium back in the blood. Right. Now what happens is, the first site where this reabsorption, most of the reabsorption takes place, reabsorption matlab we have to take it from here back into the blood. Right, that is reabsorption. And the most important site for the reabsorption process is proximal convoluted tubule. Now, proximal convoluted tubule, suppose here is the cell which is present in the proximal convoluted tubule, and here we have the blood vessel. Right, here we have the blood vessel. Now, we have to take this sodium. Now, suppose here is the sodium and here is the glucose. Now we have to take this sodium and glucose back in the blood, but in order to take it back into the blood, we have to take it through this cell. We have to take it inside the cell, and then from the cell, we have to take it back into the blood. Right. Now, here is this transporter protein on this luminal membrane. Transporter protein on the luminal membrane of this proximal convoluted tubule cell, and this transporter protein is called as sodium glucose cotransporter. Now, what did I tell you? That on every cell of the body we have that sodium potassium ATPase, right? That sodium potassium ATPase actually is present in almost every cell of the body. So definitely this sodium potassium ATPase will be present here in this cell as well. So what it does is, suppose here is the sodium potassium ATPase. What it does is it sends 3 sodium ions out. It brings 2 potassium ions in. Right. So what it actually does is it decreases the sodium concentration inside the cell. Right, by continuously sending 3 sodium ions out of the cell. Right, अब ये sodium concentration inside the cell is low and sodium concentration here in the lumen is high. What does that mean? That we have to take sodium from lumen into the cell from high concentration to low concentration. Right, but here when we talk about glucose here in the lumen is in low concentration. Glucose in the lumen is in low concentration and glucose inside the cell is in high concentration. So what does that mean? We have to transport sodium from high concentration to low concentration, that is substance A, I've talked about here. And then substance B, in this case, is glucose, and that glucose has to be taken from high concentration to low concentration. Now, what this transporter protein does is, it takes sodium from high concentration to low concentration, but as it takes sodium from high concentration to low concentration, which does not require ATPs directly, it tells the glucose that I am taking the sodium inside, let me help you as well to go inside from low concentration to high concentration. So ये transporter protein क्या करता है? It is very simple. ये transporter protein क्या करता है? ये sodium को from high concentration to low concentration लेके जाता है, लेकिन ये साथ में और एक substance को भी लेके जाता है from low concentration to high concentration, and that another substance in this case is going to be glucose being transported from low concentration to high concentration. Right. Now, where was the ATP utilized? ATP was utilized here by the sodium potassium ATPase. If this sodium potassium ATPase is not utilizing energy, this sodium potassium ATPase is not working. If sodium potassium ATPase is not working, that means we can say sodium glucose cotransporter cannot work as well, because in order for sodium glucose cotransporter to work properly, we want sodium potassium ATPase to work properly, because जब तक sodium potassium ATPase सोडियम को बाहर नहीं भेजेगा, हम सोडियम की कॉन्सेंट्रेशन सेल के अंदर कम नहीं कर पाएंगे. अगर सोडियम की कॉन्सेंट्रेशन सेल के अंदर कम नहीं है तो सोडियम को हम मतलब अगर ये कम नहीं है, ज्यादा है, तो हम यहाँ पे सोडियम को ट्रांसपोर्ट नहीं कर पाएंगे. अगर हम सोडियम को ट्रांसपोर्ट नहीं कर पाएंगे तो हम ग्लूकोज को भी ट्रांसपोर्ट नहीं कर पाएंगे. Right? So this sodium potassium ATPase is utilizing ATP, and it is utilizing ATP so that the sodium glucose cotransporter can work properly. That is the first example of the secondary active transport, and that is called as sodium glucose cotransporter. Now what it does is it sends the sodium inside the cell, and definitely sodium is being transported from high concentration to low concentration, and it also transports glucose into the cell. But remember that this glucose is being transported from low concentration to high concentration. Right, that is the first example. But remember, at the same site, there is one more transporter protein there working with the same mechanism, that is sodium amino acid transporter. Right, co-transporter. And sodium amino acid co-transporter, what it does is it takes the sodium from high to low concentration, but it takes the amino acids from low concentration to high concentration. Sodium being transported from high to low helps the amino acid to be transported from low to high, again in the proximal convoluted tubule, in order to be reabsorbed back into the blood circulation. Right? So that is all about the sodium glucose co-transporter. Now, the second is, what did I tell you, is that it is not necessarily two substances only being transported. There can be three substances as well. And one more thing is that if the substances are being transported in the same direction, as in this case, sodium and glucose are being transported in the same direction, so it is going to be sodium glucose symporter. Symporter means when two substances are being transported in the same direction. So this is actually going to be symporter. Right? Symporter is when the two substances are being transported in the same direction, and when these substances are being transported in the opposite direction, they are called as antiporter. We will talk about the example of antiporter as well. Right. Now, the second example we are going to talk about is again there in the nephron. Okay. So nephron mein we have another part. After proximal convoluted tubule, we have the loop of Henle. Right? And in loop of Henle, we have a special transporter protein there that is called as sodium potassium 2-chloride co-transporter. Now, sodium potassium 2-chloride co-transporter, very simple, name indicates that it is going to be transporting one sodium ion, it is going to be transporting one potassium ion, and it is going to be transporting 2 chloride ions. Right? Now, the thing is, whether sodium, potassium, and 2 chloride ions are being transported from outside to inside the cell, or they are being transported from inside to outside the cell. Now, what happens is, now suppose here is loop of Henle, and in loop of Henle, we have the descending limb of loop of Henle, and then we have the ascending limb of loop of Henle. And in the ascending limb of loop of Henle, we have here the sodium potassium 2-chloride co-transporter. Now, what it does is it sends one sodium ion out in the interstitial fluid outside the cell, and it is sending one potassium ion outside the cell, and it is also sending 2 chloride ions outside the cell. Right? All these are being sent outside the cell. Now, definitely sodium from high to low concentration, chlorine from low to high concentration, and the potassium is being transported from low to high concentration. Sodium and chlorine is being transported from high to low, and potassium is being transported from low to high. So, in order for potassium to be transported from low to high, what we do is we take sodium and chlorine from high to low, and sodium and chlorine are helping the potassium to be transported from low to high. Right? So, that is from lumen of this ascending limb of loop of Henle into the cell. Now, suppose it is, don't get confused here, suppose the cell is here. So, we have to take from lumen into the cell, one sodium ion, one potassium ion, 2 chloride ions. Now, outside the cell, sodium is in more concentration, chlorine is in more concentration, and potassium outside the cell is in less concentration. So, sodium from high to low, potassium from low to high, chlorine from high to low. Right? So, that is sodium potassium 2-chloride co-transporter. Right? Now, what is the importance of this sodium potassium 2-chloride co-transporter is that when we are sending all these into this interstitial space here or interstitial fluid here, what we have to do is this interstitial fluid actually becomes more salty, that because there is more salt present now there. We are actively actually by the mechanism of secondary active transport, we are sending sodium there, we are sending chloride ions there. What it does is it makes that area very salty. And we have talked about in the facilitated diffusion that the first example of facilitated diffusion we have talked about was the osmosis, where the water was being transported from low salt concentration to high salt concentration. And if this is high salt concentration here, water in the descending limb of loop of Henle will move out in this area. Right, so that is actually for the movement of water, we are making that area more salty. We have to talk about this in more detail in the formation of urine, inshallah. So अभी आपको याद रखना है that this sodium potassium 2 chloride co-transporter actually follows the secondary active transport mechanism, where one जो है substance is being transported from low to high concentration and the other substances are being transported from high to low concentration. As we have talked about the proton pump, there is importance of this transporter as well. We have a special drug called as diuretics, and diuretics mein we have a special class there which are called as loop diuretics, and those loop diuretics are actually working against this transporter protein there. They are blocking this transporter protein, and when this transporter protein is blocked, that means we are not sending sodium and chloride out in the interstitial fluid. If sodium and chloride is not sent there, this area will become less salty and there will be less water going out, and we are eliminating more water out of the body. That is particularly used when there is, for example, when there is edema, when there is accumulation of fluid inside the body, somewhere in the body, and when there is, for example, hypertension ke liye what we do is we want to decrease the fluid volume inside the body or blood volume. Now that fluid is being excreted more from the body. Uske liye, what we have to do is we have to block this protein here. If we are blocking this protein, sodium and chloride cannot go outside. Sodium and chloride cannot go outside. Water cannot move out. Water cannot move out. That means water will be eliminated through the urine. That means more water will be eliminated through the urine. That is the loop diuretics working there in that area. Right. Now we have another thing that is actually called as sodium calcium exchanger. Right, that is sodium calcium exchanger, and that sodium calcium exchanger actually is particularly present there in the that is present in the muscle cells. I'm very sorry, I forgot that. So that is present in the muscle cells. So we have the sodium calcium exchanger. Now what happens, now suppose this is the muscle cell membrane. Now what happens is the sodium calcium exchanger, it actually transports sodium and calcium, but it is exchanger. It is also called as sodium calcium antiporter. So what it will do is it will transport sodium and it will transport calcium. But remember, one substance has to be transported from low to high concentration, and one substance has to be transported from high to low concentration. Now, high to low, it is again going to be sodium being transported from outside to inside, and definitely calcium is also in higher concentration outside the cell, but in this case, the calcium has to be transported from low concentration to high concentration. What does that mean? It is going to be transporting sodium into the cell down the concentration gradient. That means from high concentration to low concentration, right? From high concentration to low concentration. But this transporter protein is going to be transporting calcium from low concentration to high concentration. It is very important that in the muscle cells mein, what we want is we want to make that balance of the calcium inside the cell. So uske liye jo hai hum calcium ko inside of the cell to outside भेज देते हैं और हम sodium को from outside हम inside भेज देते हैं. Now again, there is a drug called as digoxin. Digoxin is actually positive ionotropic agent that actually increases the contractility of the muscle cell. Okay, of the cardiac muscle cell particularly. Cardiac muscle cell ke liye digoxin is given, and that digoxin, what it does is it increases the contractility. Now how it increases the contractility is it actually blocks the sodium potassium ATPase. And if it blocks the sodium potassium ATPase, what does that mean? That it cannot send sodium ions out, it cannot bring potassium ions inside. If we are blocking the sodium potassium ATPase, what did I tell you? That all the secondary active transport mechanisms are actually depending on sodium potassium ATPase. Sodium potassium ATPase is working, they will work properly. This is not working, they will not work properly. And if sodium potassium ATPase is blocked by digoxin, what will happen? That this transporter protein cannot work, and if it cannot work, we will accumulate more calcium inside the cell, and if we are accumulating more calcium inside the muscle cell, that means definitely there is going to be increased contractility. So increased contractility ke liye digoxin actually decreases the activity of this transporter protein, but it does not decrease the activity of this transporter protein directly. What it does is it decreases the sodium potassium ATPase activity, and once we are decreasing the sodium potassium ATPase activity, we are definitely decreasing the activity of sodium calcium exchanger. Now, let us finish the secondary active transport with the last example, and that last example is going to be sodium proton transporter. Now this sodium proton transporter is again going to be the best example for this is again going to be the renal tubules or kidney tubules or the tubules of the nephron. And most importantly, for example, it is there in the collecting duct or in the distal convoluted tubule. What is there is most importantly the collecting duct. Now collecting duct mein what happens is, suppose here is the lumen of the collecting duct and here is the cell. Now what happens is that, for example, our हमारे collecting duct में we have intercalated cells, and those intercalated cells are actually involved in the regulation of acid-base balance, right? That is involved in the regulation of acid-base balance. And yeah, now what happens is that here is that transporter protein, and that transporter protein is sodium proton cotransporter, and what it does is it will definitely send the sodium from lumen into the cell, and we are actually excreting the hydrogen ions or these protons out of the cell. But these protons are being transported from low concentration to high concentration, and the sodium is being transported from high concentration to low concentration, right? So we have the sodium potassium I'm very sorry, it is sodium proton cotransporter, and this sodium proton, for example, we have acidosis. Okay, we have acidosis, and that acidosis means there is more hydrogen ion concentration there in the body or there in the blood, right? So what we want is we want to excrete these hydrogen ions along with the urine, and one of the best organs in our body maintaining this acid base balance is going to be our kidneys. So kidneys, अगर वहां पे acidosis है, that means hydrogen ion concentration ज्यादा है, तो hydrogen ions को excrete किया जाएगा. और अगर हम जो है हमारी बॉडी में alkalosis है, that means hydrogen ions हमारी बॉडी में और चाहिए to neutralize those या to increase that, to decrease that pH. So hydrogen ion concentration वहां पे ज्यादा करने के लिए we are actually reabsorbing those hydrogen ions from these intercalated cells into the blood. And if we have acidosis, तो उस कंडीशन में हमें हाइड्रोजन आयंस को जो है हमारे यूरिन के साथ एक्सक्रीट करना है, because we already have more hydrogen ions there and we are reabsorbing bicarbonate ions. So hydrogen ions को excrete करने के लिए there is a transporter protein there, but hydrogen ion has to be transported from low to high concentration. But in order for this hydrogen ions to be transported from low to high, it is actually helped by the sodium, when sodium is being transported from high to low, and this transporter protein helps the protons or hydrogen ions to be transported from low to high. Right? So that was the last example of secondary active transport. Now we have talked about the active transport in this class, and the active transport, we have talked about two mechanisms. One is the primary active transport and second is the secondary active transport. Primary active transport utilizing the ATPs directly, and secondary active transport utilizing the ATPs but indirectly. And in both, there can be the transport from low concentration to high concentration. But remember, in primary active transport, there will be transport always from low to high concentration. And in secondary active transport, there are more than one substances being transported. And one is mostly going to be sodium, but that sodium is being transported from high to low concentration. And another substance which will be transported along with the sodium will be definitely from low concentration to high concentration. And second thing is that in the secondary active transport, if the substances are being transported in the same direction, that is going to be called as symporter mechanism. And if they are transported in the opposite direction, for example, sodium is going inside and another substance is going outside, and that is going to be the antiporter mechanism of secondary active transport. That is all for today. See you in the next lecture. Allah Hafiz.